Well, good evening, friends, and thanks for joining us tonight. This is John, and our topic tonight is how to manage microbial biostimulants. So, when we have a conversation about microbial biostimulants and how to fit biostimulants into an overall agricultural ecosystem and a farm management system, there are a lot of questions today about how to fit biostimulants into an integrated agronomy system and how to position them to get greatest crop response. And this is a conversation dialogue that I personally find very intriguing. Over the last 10 years with our work at Advancing Eco-Agriculture, we have done a lot of work with biostimulants, and it's become a very natural process for us to think about how we integrate them and how to position them to get the greatest crop response. So we've been working with biostimulants for a very long time, and as biostimulants are becoming more adopted by mainstream production agriculture, we've discovered that many growers have a lot of questions about what is happening and integrate biostimulants to get the greatest crop response. So our discussion tonight, what I want to talk about is to help give you a framework and a thought process framework to think about how to position biostimulants on your operation to get the greatest economic crop response and to really integrate them with the other things that you are doing and make sure that you don't sabotage your efforts in the process. So I'm going to start a screen share and share a presentation with some of my thoughts. So the key, when we think about managing microbial biostimulants, the microbial biostimulants are, and just as well as plant biostimulants and biostimulants in general, are these very intriguing materials that often can have synergistic interactions or can have very intensely. Of course, when we think about, when we ask the question of what is a biostimulant, there's today a very broad-ranging definition of what defines a biostimulant. So they can be anything from weed materials, humic substances, amino acid blends, and essential fatty acids. There's this broad array of hundreds, actually even thousands of compounds that can be considered to be biostimulants, both microbial biostimulants and plant biostimulants. And so when you consider this broad array of both naturally occurring compounds as well as synthetic compounds, and you start wondering about how they could possibly interact with each other to produce the greatest crop response, it can become very complicated very quickly. And yet, at the same time, from our observation, from our experience, it can be really simple to put these pieces together as long as we approach them from the right framework. So I think the key question that we need to ask is, how can we achieve exceptional product performance? How can we get the greatest crop response from any products that we might apply? And I think the key to defining, when you ask, well, what defines exceptional product performance? The key is, I believe, that we need to manage for economic results. So one of the interests that I've had personally, and one of the things that we've seen a lot at AEA is paying very close attention to how we can produce the greatest economic response. And I believe that we integrate biostimulants with our synthetic nutrient applications or historical fertilizer applications. The combination of the two is when we partner them correctly and we can put together an integrated system where we are integrating biostimulants with typical nutrient applications and produce the greatest economic crop responses in those ecosystems. and I want to describe the framework that we use and the thought process that we use to put these pieces together. So there are a few fundamental concepts that are important for us to think about right at the beginning. And the first one is that biostimulants perform best when plants are supported with the proper nutrition. And this is a very, very important framework where we sometimes are, um, we sometimes participate in a dialogue where there is this idea that we don't have to pay as close attention to nutrition, calcium and potassium, calcium, magnesium, sulfur nutrition is important, but if we are limited on a farm economically and we're not able to address some of the major nutrient imbalances, we can just put on these biostimulants. We can put on these microbial inoculants, or we can put on these biostimulants, and they're going to stimulate plant growth and plant development. And we can overcome, that will allow the plants to overcome some of the inherent nutritional imbalances. We have not found that to be true in the field. Instead, we find that we have plants that are really well balanced from a nutritional perspective, and growers are already paying attention to calcium and magnesium and sulfur and making sure the plants have good mineral nutritional integrity. And we then combine that with biostimulants. We add biostimulants on top of the nutrition. That is when we get the greatest responses. We've had many growers who have used biostimulants in the past without paying attention to the nutritional foundation, and they report that they're not getting the crop response that was promoted by the manufacturer of the biostimulant. And in our experience, the foundation is that we need to have good mineral nutrition, and then when we add biostimulants on top of that, we get the greatest possible crop response. So this is a very, very important piece that I think is often missed by manufacturers and people who are promoting the use of biostimulants. I think biostimulants are very powerful tools, can be very effective tools, but they need the right environment and they need the right foundation in order to get the best performance out of them. So when we think about using microbial biostimulants, we think about positioning these products within an integrated system in a framework where we want to position these materials relative to three specific segments. One is we want to position them relative to the management pattern of how the overall crop is being managed. And I'll describe in a moment what I mean when I say management pattern. Second, we want to see how these biostimulants are positioned relative to other products, perhaps pesticides, fungicides, insecticides, or to other fertilizer materials that are in the system. And third, we want to position these biostimulants relative to the crop development stage and where the crop is in terms of reproductive development, if it's a reproductive, and how we position that for the greatest crop response. Because we have learned that there are, for many different crops, there are specific windows that are critical points of influence where a biostimulant application, or for that matter, any product application, can produce a tremendous crop response. So when we look at each of these three pieces, for the rest of this discussion tonight, I'm going to be talking about each of these three pieces and how we position products relative to each of these categories. And if you have any questions, comments, things that you would like for me to discuss, you can type in your question down below. You'll see a little green button in the Q&A box. Type in your questions at any point, and I'm going to quickly move through the framework that we have put together, and then I'm going to open it up for Q&A, and we'll talk about any questions or comments that you might have, as I'm sure some have some really valuable input. So let's look at each of these three pieces. The first is the management pattern. So what is a management pattern? As we've worked with biostimulants and microbial inoculants and work with growers who are managing disease and insect pests with biocontrols and trying to reduce pesticide use, we've observed that the most successful growers use a very different management style for how they think about potential pests, potential pathogens, how they can prevent them from happening in the first place, compared to many mainstream growers. So I've been thinking about how we can describe the different management patterns that these various growers and farm managers used. And I discovered a management pattern outline that was first developed for lean manufacturing and Six Sigma that I think is very relevant to describing how growers work with cues and signals that they're getting on their farm. So this is a management pattern that is referred to as systems analysis for thinking about the entire operation, the entire farming operation as an integrated system. So the first management style is what is referred to as being reactive. So when we are managing reactively, we are managing things that have already happened. So perhaps we're looking at the crops visually and we're looking for visual symptoms of nutrient imbalances. So we're really managing in the past. We're managing what has already happened. And the alerts, when we get an alert about something that is going wrong, such as we have a pathogen infection or we have a spider mite infection in the crop, the alerts occur at the point of failure. So it's already happened, and that's when we first find out about it. So in that type of management pattern, we are dependent on rescue treatments by using cleanup pesticides. So using very strong insecticides or fungicides that have the capacity to clean up a problem once a problem has already happened. The second management pattern is referred as proactive management. So proactive management is working within the larger environment of things that are happening around us and trying to manage what is happening in the present moment. So the alerts will occur before the actual point of failure. So perhaps we're working with a crop scout who might notify us that there are spider mites in the region, in the area, and even though we don't have spider mites on our farm yet, we know that they're likely to be there at some point. And so we are able to use preventative treatments using biocontrols or using softer treatment modalities to try to prevent problems from actually showing up in the first place. And then there is the third level of management, which is referred to as predictive management. So predictive management is towards the future, where we know that we're growing a particular crop in a particular environment that is susceptible to spider mites, and we have spider mites present in the crop 4 years out of 5. So we know that it's very probable that we will have spider mites in our crop again in the future, and we want to prevent that. We want to prevent spider mites from ever becoming a problem. I'm just using spider mites as an example. So our goal with predictive management is to prevent the root cause of the problem from ever becoming a problem in the first place. So if we continue with the spider mite analogy, our goal is to try to understand why the spider mites are attracted to a given crop and change that crop's nutritional profile or change the environment in such a way that we remove the potential for an infection. So this is the framework where biostimulants really fit, because we can use biostimulants to change a crop's nutritional profile or to change a crop's energy so that it can be much more resistant to diseases that might show up in the future. And so I want to describe a little bit how that can fit. So my point here is when we think about positioning biostimulants relative to management patterns, when a grower or a farm manager is managed very reactively and is in constant reactive mode to challenges that are happening, disease control and pest control and so forth, then it is much more challenging to get a strong economic crop response using biostimulants. If we start moving into proactive management and predictive management, where we're trying to identify and address the root causes of problems, where biostimulants produce the greatest crop response and where they're the most valuable. So this is an important thought process. This is, of course, dependent on the farm manager, but really successful farm managers will use biostimulants to try to prevent problems long before they occur. and to address root causes. So what we have observed when we work with all the various farm managers that we work with growing a broad variety of different crops is that the most successful and the exceptional managers are those who first try to identify the root cause and prevent problems from ever happening in the first place. And only as a last resort will they try to use chemistry and pesticides as needed. So the second thought process that we think about how to manage or how biostimulants is relative to other products. And this is very important. In fact, it's perhaps the most important part of this entire discussion. So we've found that biostimulants can have very strong, very exceptional synergistic effects with other biostimulants or with other nutrient products, or they can have very antagonistic effects. And so I want to just highlight some of the most important and most influential synergistic and antagonistic effects that we have found. So we find that many biostimulants have very strong synergistic effects with other biostimulants, and that we can stack different products together to get a maximum impact. For example, when we stack seaweed, which might contain cytokinins and ox and a number of other compounds that stimulate and enhance plant development, with PGPRs, which is plant growth promoting rhizobacteria, with mycorrhizae. So here we're combining a seaweed product with a bacterial inoculant with a mycorrhizal product. When we stack those materials together, we produce a much greater response than either of those products individually. It's a compounded response. So it's almost a little bit like using a black box mathematical equation where I've described some of the effects that we've observed on plants is that 1 + 1 doesn't always equal 2. Sometimes with these biostimulants, 1 + 1 can equal 11. And it's a lot of fun when that happens, when you apply small amounts of biostimulants and you produce this tremendous compounded crop response that is much bigger than you expected. It's a lot of fun. Lots of amazing things can happen with plant development and plant growth and yield and quality when we achieve those results. So how do we do that? So I want to give you examples of synergistic materials that we have worked with and that we've seen. So we have learned that there are very strong synergistic effects when we combine mycorrhizal fungi and trichoderma. So mycorrhizal fungi, I'm sure many of you are very familiar with them, colonizing living root systems and being able to absorb water and various nutrients, particularly phosphorus, and access them. Even plant roots can't access them, and having a symbiotic relationship with plant roots. And trichoderma are known for developing a very disease-suppressive rhizosphere and strongly antagonizing a number of potential soil-borne pathogens like Phytophthora and Rhizoctonia, and Pythium, Verticillium, and Fusarium, and so forth. And we find that when we combine the two of these inoculants and we apply together, we get a much greater response than we do with either one of them alone. We see a similar effect when we use SAR triggers. So products that are SAR triggers, SAR stands for systemic acquired resistance. And there are a number of biocontrol products and as well as biostimulants that trigger effect. So if you think of chitosan, for example, or the chitinase enzyme as well, and to some degree chitin also, are known to trigger the jasmonic acid pathway, which is the systemic acquired resistance pathway in plants. So they're triggering this immune reaction. And there are also many of the PGPRs, plant growth promoting rhizobacteria, that have this same effect where they trigger a SAR response. So when we apply a product that triggers this SAR response and we combine it with seaweed, which has a variety of different compounds that have a synergistic effect with the SARS, we actually get a much bigger immune response in the plant. So for example, one example of this is a farm that we were in central Pennsylvania, perhaps, I don't know, 5 or 6 years ago. And this was on row crop organic corn, food grade corn, about 2 weeks or so after germination. So the corn seedlings were about V3 to V4 stage, anywhere from 8 to 12 inches tall. And this farmer, this entire field had a severe problem with corn rootworm and wireworms. So the farmer, this was an organic farmer, he didn't have the ability to put on any pesticide applications, and he wanted to know if there was anything that he could do to allow the plants to overcome and overpower and outgrow the corn rootworm and the wireworms. So we made a recommendation for a combination. There were 3 products. One of them was an AEA product called Photomag. The second was a product called Sea Shield, which is essentially a liquefied crab shell and shrimp shell. We have the chitosan effect, which is a SAR trigger. And the third material was Sea Stem, which is a seaweed product. So we have seaweed, chitosan, and Photomag, which contains a very strong, it's a mineral nutritional supplement that contains magnesium and sulfur and cobalt and molybdenum and boron. And I feel that of particular importance in that combination were the molybdenum and boron. So here was a combination of a nutritional support, a chitosan material that would trigger a SAR response, and seaweed. And I told the grower when we made this recommendation, I told him I have no idea whether this is going to work or not. We've not tried it before. We don't know whether it's going to be successful. So they put on a foliar application of this material, and 48 hours later, the corn rootworm larvae and the wireworms in the root system were dead. There was such a strong compounded effect that the plant had such a strong immune response in the plant that the plant actually produced compounds that killed the corn rootworm and the wireworm. And that's a lot of fun. It's a lot of fun. These are the types of effects that we have seen all the time, is that we can actually use these biostimulants to produce such a strong immune reaction within the plant. We do disease resistance and insect resistance. And this is one of the pathways that we have seen be very effective. So other examples of synergistic materials for biostimulants would include bacterial inoculants and fungal inoculants. So when we combine an inoculant which is primarily bacterial, such as the plant growth promoting rhizobacteria, and we combine that with fungi, such as mycorrhizal fungi or Trichoderma, it's unfortunate that many of the products that are available in the marketplace today are only one or the other. They're either only a fungal inoculant or only a bacterial inoculant. And we find that when we combine the two and put both together into the same application is when we get the greatest crop responses. And we've developed a number of our own materials, such as BioCoat Gold, using this principle. Other examples of synergistic materials include microbial inoculants, either fungal or bacterial inoculants, combined with humic substances. So using humic acids and fulvic acids can increase the microbial inoculant response to an even greater degree. And one very large area of biostimulants that holds a lot of promise, it already is well understood to some degree, but there is still a lot of potential, I believe, is with using phytohormones and more deeply understanding cytokinins and auxins, gibberellic acid, and so forth. There's a lot of untapped potential in the use of cytokinins and auxins. And it is fascinating to me how these plant hormones can have such intense synergistic effects with the proper nutritional support. So, for example, just a very brief example is the synergistic effect of applying cytokinins with calcium and cobalt. So when we apply a cytokinin product, let's say for the sake of discussion, a seaweed that is known to have a specific concentration of cytokinin, such as 400 parts per million for the sake of discussion. If we apply that cytokinin product by itself, we can produce, let's say we're growing a tomato crop and we want to produce a very strong reproductive response and trigger flower formation and a lot of new bud development. If we put on a cytokinin application with seaweed at a rate of 2 quarts per acre, we might increase the bud content on the tomato crop by 30%. I'm just making up a hypothetical scenario based on some of the observations that we've had. However, when you combine that seaweed product and the cytokinin application at the same application rate of 2 quarts per acre with calcium and cobalt, let's say we add a quart of liquid calcium and a small amount, let's say a couple ounces or a pint per acre of a cobalt application, of course, is all dependent. You might get a 60% or an 80% increase in bud development. So there is, and quantity of flowers. So there is the tremendous additional response when we combine plant hormones with the right nutritional support. And this also goes back to, which was that these phytohormones and many of these materials, actually these biostimulants, produce an even greater effect if and when the plants have the needed and the necessary nutritional support to begin with in the first place. So now you can produce an even greater crop response. So if you think about all these various major synergistic materials that we use, but the way that we think about combining these biostimulants is to develop the term synergistic stacks, where we stack products in these various categories together to produce the greatest response. So if we stack and combine a bacterial inoculant with a fungal inoculant, and then we add a microbial biostimulant, which would combine, which would stimulate both bacteria and fungi. This could be an enzyme product. It could be humic or fulvic substances. There's many things that could serve as a microbial biostimulant. And you combine with a plant biostimulant, such as seaweed or a phytohormone, and you combine that with plant nutrition, such as calcium or cobalt or whatever is needed by the plant at that stage of development. When you combine all of these things together into the same application, the same sprayer solution or the same fertigation, you get this tremendous crop response. And this is truly where 1 + 1 can equal 11, where you get this tremendous root development and plant development that you don't see when you apply any of these materials on their own. So let's also speak for a moment about some of the antagonistic materials. So the antagonistic materials that we've seen that do not work well together at all is when we combine, one example is when we combine mycorrhizal fungi with soluble phosphorus. So if we add soluble phosphorus product, such as liquid phosphorus 989, for example, to a mycorrhizal fungi application, the effect will be that it will suppress and almost completely shut down the mycorrhizal fungi, and they will not establish a viable colony and will not colonize the root system. So that is one combination that doesn't fit together at all. Another example of antagonistic materials is products which produce a SAR trigger effect, such as the chitosan that I was referring to earlier, or plant growth promoting rhizobacteria, or products such as regalia, for example. Japanese knotweed extract produces a SAR trigger effect. Those materials do not work well when plants have high levels of nitrates in the plant sap or when they're combined with a nitrogen application. So if there are high levels of nitrates in the plant sap and plants have a surplus of nitrogen, these materials don't produce nearly the crop response. The plant is expending a lot of energy in trying to deal with the nitrates. Another example, similar to mycorrhizal fungi and phosphorus, is azotobacter and soluble nitrogen. So we find that when we have high levels of soluble nitrogen present in the soil and we add azotobacter bacteria, which is nitrogen. So this is actually a very interesting point. Today, with all of the farms that AEA works on throughout North America and throughout the world for that matter, the thing we have experienced consistently for a long time is the farms that we work with practically never experience a nitrogen deficiency. In fact, one of the challenges that we deal with regularly is excess nitrogen in the plant. Whether we're talking a tomato crop or salad greens or tree fruit or nuts or whatever the case might be, we're constantly, or even commercial corn production, we're constantly monitoring and trying to manage to make sure that we don't end up with excessive levels of nitrogen inside the plant. And we've been able to produce this response. This is almost entirely coming from bacterial inoculants, which include Azotobacter and other bacteria, which have the capacity to fix nitrogen for a crop, even in the absence of legumes. So they don't have a symbiotic relationship with legumes. They're able to fix nitrogen and supply nitrogen to any living crop. And so I just realized recently I was giving a presentation about biostimulants and biocontrols when someone asked me this question, and I realized that, well, actually, the question was brought up that other growers aren't experiencing this same level of success as Azotobacter. They haven't been able to use bacterial inoculants to supply a substantial amount of nitrogen. And I believe the problem is not with the bacterial culture, with Azotobacter, because we have dozens of farms that we point to where we have where the crop has a generous supply of total nitrogen, where there is no applied nitrogen. We have, just 2 years ago, I was on a farm in eastern Pennsylvania. It was organic corn production. So there had been a cover crop planted and incorporated prior to planting the corn crop. But this was a corn crop that produced 320 bushels per acre of corn production with no added nitrogen, not even in the form of liquid fish or anything like that. There was zero added nitrogen. It was 100% coming from cover crop and the microbial inoculants. And the reason for that, I believe we've been able to see success where other farms have not, is that we have combined these bacterial inoculants with other biostimulants that enhance their effect, and we have not used any of these antagonistic materials such as soluble nitrogen. So that's something that I can expand on later if any of you have any questions about that. An additional organizing microbials and oxidizing fertilizers. So whenever if you have a salt fertilizer solution, and I refer to oxidizing when you think about it, let's say you have a salt fertilizer solution and you have a little cut on your finger, you get some of the fertilizer into a little cut, it starts burning. That burning is oxidation of the cells. And the exact same thing happens to bacterial cells that are in the solution or that are in the soil profile. So we find that many fertilizers such as ammonium sulfate or urea or many of these soluble ionic fertilizers and nutrient solutions have a very strong antagonistic effect to microbial inoculants. So we're not suggesting that we should not apply these materials, but I would only suggest that they need to be buffered out. They need to be buffered with humic substance or with carbohydrates or other materials to buffer them out so that they don't have such a strongly antagonistic effect on the microbial population rises here. And then another very important and very obvious antagonistic material combination is the use of phytosanitary bacteria for control of potential bacteria and fungal pathogens on the leaf surface, on the phytoplane, combined with bactericides. So there are a number of biological inoculants combined with enzyme biostimulants available today. One material that we use a lot is Micro 5000, which is a blend of phytosanitary bacteria that colonize the leaf surface and have a symbiotic relationship with the plant that is very similar to the symbiotic relationship that bacteria and mycorrhizal fungi have in the root system. So we have this symbiotic relationship happening on the leaf surface. And obviously that microbial population population is going to struggle and perhaps even disappear completely when we spray the plant with bactericides, such as copper applications or manganese applications, or peroxide, hydrogen peroxide, or ozone, for example. So I would say, and again, I'm not saying that we should never use copper or manganese applications. If we have challenges with bacterial diseases, there is an appropriate time and a place for those treatments. I am suggesting that, I would suggest that we can get to a point where we are using other tools and we're no longer dependent on copper applications and so forth. But in terms of positioning biostimulants and positioning these biological products, they can best be positioned in crops and in areas where they don't conflict with the bactericides that are being applied. That's the only thing that I'm suggesting. And then continuing that theme, of course, fungal inoculants and fungicides. These are an obvious oxymoron to apply these two materials in combination with each other or in close relationship to each other in terms of timing. So the third piece that I'd like to discuss is how to position biostimulants relative to crop development stages. And this is an intriguing discussion. Something that we've observed is that for many crops, there are these very narrow windows that we call critical points of influence, where a product, whether it's a nutritional product or a biostimulant, whatever the case might be, when we apply products within this specific window of application, we get this tremendous response, much greater than we do at other times. So we've tried to identify what these windows are and where they occur. And this is a topic for a whole other webinar at some point. But I want to describe briefly, at least on a very high level, so you get an idea of the work and the thought process that we use to identify where we can position these materials. So all plants, I should say all reproductive plants, where we're actually selling the fruit or the seed, go through this continuous growth cycle where there's this extension contraction cycle, or vegetative versus reproductive growth, or male versus female energy. There's many different terms that we can use to describe it, but perhaps the most common and best understood would be when we talk about vegetative growth versus reproductive growth. So there are these constant cycles and switches that happen back as plants grow. So if you think of a tomato plant, for example, you begin with a very small tomato seedling. There's a tremendous amount of energy that is packed into the seed. And then that energy, as the plant begins to expand outward, you get the seedling, you get vegetative growth, energy becomes more diluted. And the small tomato seedling initiates a bud and starts putting a bud together. You have this concentration of energy into the bud. So that's the contraction phase. So you start with vegetative growth, which is expansion. Then you go to bud development, which is a contraction of energy. And then when that bud blossoms and pollinates, you now go to the fruit period, where again you have this expansion of energy. And then as the fruit matures and you start at seed maturity, now there's this concentration of energy again, where energy is concentrating in the seed. This happens for all of the reproductive plants throughout the plant's life cycle, where there's this constant switch. So what I'm attempting to describe is to describe which of these energy patterns is dominant in the plant. So there is not, so the important point here is that both reproductive and vegetative energy and vegetative growth are happening in all plants at the exact same time. It is never exclusively 100% one another. It's more like their balance, where one is 49% and the other is 51%. And this pendulum effect, where they're constantly swinging back and forth, where first one is dominant and then the other is dominant. So let's look at what some of this might look like for various crops. So if you think of a corn plant, how many cycles, how many switches from vegetative to reproductive would you anticipate that a corn plant might have? when growers sometimes tell me that, um, well, they would expect a corn plant to have one switch, one cycle, where it's going to be it's going to be viv from the moment the seed germinates until we get to the tasseling and silking stage, and then it's reproductive for the rest of its life cycle until all the way through until harvest. And that certainly, it might appear that way from a macro perspective, but when we look at what is actually happening with hormone profiles and what is happening inside the plant, actually 13 cycles that happen. So this diagram is an outline of all the various cycles that are happening and what stage plants are switching from vegetative growth to reproductive growth. So at V1, shortly after germination, obviously we have vegetative growth, and then at V2, it switches to very early bud initiation, or the ear, where the ear embryos are determined at the V2 stage. V3, it switches back to vegetative growth, and then V6, it switches back to reproductive dominance, where the number of rows on each of the on each of the ears is being differentiated and being determined. And so you continue throughout this entire life cycle for corn plant, where you have 13 transitions, where it's switching back and forth from one to the other 13 times. So this same pattern holds true for many of the various crops that were for all productive crops. So if we're working with spur-bearing trees, we have 6 of these macro trends where we're switching from bloom pollination to shoot initiation, back to bud initiation, fruit fill, and so forth, where we have this constant switching back and forth. So why is this worth talking about, and why does it matter? Well, what we have learned is that there are specific moments in these windows where any nutritional product or a biostimulant can have tremendous impact on yield, or they can have a tremendous impact on disease and insect resistance. And they're not the same times, they're not the same moments. There's one moment for disease and insect resistance, and there's another moment for yield potential. Nutritional products and biostimulants will affect what is happening in a plant differently at different stages. So when you have a lack of nutritional integrity, or you have any level of plant stress, at the peak moment of each cycle, you sabotage the plant's yield potential. So let's say, and this effect is greatest when you are in the reproductive cycle. So let's say, for the sake of discussion, that you have an apple orchard blossoming and pollinating. So that blossoming and pollination is the reproductive cycle. So at the peak moment of that reproductive cycle, right at the moment where the majority of the blossoms on a tree have just been pollinated, is when you have this peak, this crest. And at that peak moment, if you have any nutritional stress, lack of calcium, lack of phosphorus, boron, zinc, manganese, copper, the case might be, that nutritional stress is going to lower the yield potential by lowering the quality of pollination and bud development that is happening. So this is where you really have an impact on yield. But this is very different from the moment where you have the biggest impact on disease and insect resistance. So disease and insect resistance, any lack of nutritional integrity at the transition from vegetative to reproductive is what triggers proteolysis and creates disease and insect susceptibility. So if we continue with our apple tree analogy, when an apple tree switches from vegetative growth to reproductive growth is when we have the greatest level of stress. So if we continue, if we think about the blossoming and pollination period, so a tree generally will move into winter dormancy, and the dormancy period is generally a period where we have a dominance of vegetative energy, vegetative growth. So the tree is relatively dormant, but vegetative growth is still a dominant energy during the dormant period. And the entire tree switches from vegetative dominance to reproductive dominance when we have all this pollination happening. So it is at that moment when this switch happens from vegetative growth to reproductive growth is often when we have the greatest degree of disease and insect susceptibility. Think about, if you think about apples, think about all of the diseases and all the insect pests that have their greatest economic impact in the first month after blossoming and pollination. It's a long list. It's apple scab, sooty blotch, cedar apple rust, codling moth. Kind of there's a pretty long list that has an impact right at this stage. Why is that? This is true because when plants shift into reproductive mode, all of the reproductive processes, seed development, blossoming, pollination, so forth, the reproductive parts of the plant have the highest mineral concentrations. So a flower blossom, a blossom might have zinc and manganese and copper levels that are as high as 8 to 10 times higher than a leaf tissue or vegetative buds at the same time. So they're going to have much greater nutritional requirements. And if the plant is unable to supply the nutritional requirements for those reproductive buds or for those blossoms, it will switch, the entire plant will switch from protein synthesis to proteolysis, and it triggers greater disease and insect susceptibility. So this is a conversation that's supposed to be about positioning biostimulants, not about what is happening inside plants. It's obviously a much topic that we will discuss more in depth on point, but when we think about where to position biostimulants to have the greatest economic response, the optimum positioning is right at these maximum stress periods when a plant is transitioning from vegetative to reproductive mode. That's when we're going to have the greatest crop response. So this is what we refer to as critical points of influence, and is a bit of a description for where we can have the greatest response. So I see that I'm taking a bit more time than anticipated. I'm going to try to move through this last section fairly quickly. Going back to where we started this discussion when we talked about positioning products into a farmer's management pattern. And when we look at a question that we're often asked is, growers want to move to be in a more predictive management pattern and to understand what the root causes of disease and insect challenges are and how we can address those trending towards the future rather than just waiting for them to show up. So I wanted to describe very briefly, very quickly, and highlight some of the research that has been around for quite some time that points to understanding the root causes, the underlying foundational causes of disease and insect infestations, and how we can manage them and how we can prevent them. This is something that we've been very fortunate and very blessed to at AEA. We've had some very good mentors who have taught us some of these pieces on how to manage and how to prevent disease and insect problems, and we've been very successful accomplishing that on a large scale, which is something that I'm very, very appreciative for. We've had some exceptional successes, and it's a good feeling. It's a lot of fun. So William Albrecht at the University of Missouri in the 1940s said that insects are nature's garbage collectors and diseases are her cleanup crew. And this is the fundamental philosophy that we have advancing eco-agriculture. Is we believe that insects and diseases are simply nature's survival of the fittest mechanism. They are here to take the unhealthy plants out of the system. And that these pathogens only show up when we have plants that are stressed and that have severe nutritional imbalances, that when we correct these nutritional imbalances, we can actually prevent disease and insect infestations. And this is something we've been very successful at, and I want to describe on a very high level some of the thinking and the thought processes that we use. So there's a lot of research and a lot of discussions around plant immunity, plant immune pathways. I briefly spoke about systemic acquired resistance. There's a second pathway called induced systemic resistance, which is based on salicylic acid. what I can say very simply is that when we look at plant immune systems, there are two types of immunity. There is what I would refer to as passive immunity, and there's active immunity. So passive immunity is when we simply remove the food source. So when corn rootworm larvae are dependent on soluble amino acids and nitrate nitrogen in the plant sap. If we change the amino acid profile and we change remove the nitrate concentrations, the plant that is those root systems are no longer digestible, and they're no longer provide a food source for the corn rootworm larvae, and the larvae goes away, dies, starves, and is no longer a problem. This is true for all the insects that have simple digestive systems. And then when you have active immunity is when you trigger a SAR or ISR response in plants, and you actually have an active immune response where plants are producing compounds that can actively antagonize and even kill bacterial and fungal pathogens. So the simplest way to think about this is when we think of the photosynthesis process. I'm going to use carbohydrates as a brief illustration. This is true of both carbohydrates and proteins. So during the photosynthesis process, the sugar that is produced as the final result of photosynthesis is this simple sugar called glucose, which is C6H12O6. So visualize this, imagine this as being a short chain that has 24 links. 6 links of oxygen, 12 of hydrogen, and 6 of carbon. Then that short chain molecule of glucose is bonded together end to end. It's rearranged, it's restructured, it's added. That's the foundational building block that is used to build all the rest of the compounds inside a plant. So, I'm going to switch to proteins for just a moment. So when you think about a protein, the constituents of proteins are amino acids. So think of this short chain sugar molecule called glucose that now gets nitrogen added to it, and the structure gets rearranged into different forms and shapes. The combination of sugars with nitrogen is now referred to as an amino acid. So there are 23 common amino acids, and these amino acids are all relatively soluble in plant sap, and they move around in plants relatively easily. So then these amino acids get bonded together to form much longer chain, much more complex compounds. Well, first they're bonded together to groups of 2 or 3 that are now referred to as peptides. So you might have dipeptides or tripeptides. Then these peptides are bonded together to form much longer, much complex chains that are now referred to as complete proteins. The key is that at each step of this pathway, we have a welding crew inside the plant that is responsible for forming all of these reactions and putting all these bonds together. And this welding crew is called enzymes. So enzymes are like an end wrench in that each enzyme can only trigger one specific reaction. And just like an end wrench can only fit one size nut or bolt properly. But also like an end wrench, an enzyme can do the same job and perform the same job over and over and over again without being used up. And similarly, an end wrench can tighten hundreds, thousands of nuts. So this is important and worth talking about because this is at the foundation of the differences between the digestive capacity of an insect versus the digestive capacity of a human or an animal, for example. So we know that different animals and insects and people have very different digestive systems with very different digestive enzymes. So if you were to compare our digestive system with a digestive system of a ruminant animal such as a dairy cow, dairy cows can break down and digest cellulose because they have the cellulase enzyme in their digestive system. We don't have that enzyme, and so we cannot consume hay and forages and digest those and get our energy source from them, but cattle can because of that enzyme. This is also the foundational difference between our digestive system and the digestive system of some insects. Many of the larval insects don't have the digestive enzymes that are required to break down complete proteins. So they're not able to use plants, not able to digest plants that have complete proteins the same way that we're not able to digest cellulose. I'd like to also speak for just a moment about some of the research work that was originally done by Philip Keller at the University of Florida, Gainesville, on plant and insect communication systems. Incredibly rich research and definitely worth pursuing and reading more on if you're interested in understanding plant and insect communication. The very simple summary is that insects are communicating with environment around them, and they're communicating with plants in the infrared spectrum. And that unhealthy plants, particularly plants which have high ammonium concentrations, serve as a beacon to attract insect pests, because interestingly enough, ammonium is an infrared signal pump, an infrared signal amplifier, where Philip identified that insects depending on humidity and temperature, they can detect unhealthy plants at a distance of 1,400 to 1,700 yards. So almost a mile, they can see unhealthy plants. So what he described, it's very simplest, is that when there are healthy plants that are growing well in the field, there are no obvious disease and insects present, this is what we see. We see absolutely nothing. We see a healthy dark green plant. What insects see is they don't see anything. It shows up as being completely black on their radar screen. They can't see anything in the infrared spectrum at all. But when we have unhealthy plants, where we have insects present, we have high ammonium concentrations, this is what we see, and this is what insects see. And so you might think this is intriguing, but actually I'm paraphrasing or quoting from what I recall from reading one of Philip's books. He said that in the infrared spectrum, unhealthy plants appear to insects as a neon light against a dark background. They stand out very strongly. So if this is one of the challenges with using insecticides and fungicides, but let's say insecticides for this discussion. We can apply a synthetic insecticide and eliminate a potential pest in the field. But as long as we don't address the underlying root cause of why that insect was attracted to the field in the first place, you can clean up the insect, but you still have the sign. You still have the neon sign. So you actually have to change what is happening in that plant physiology from an underlying perspective in order to really resolve the problem from a long-term perspective. So this is the bottom side of a tomato leaf at 4,000 times magnification, showing all the trichomes that are vibrating at 36,000 megahertz. These are photon receptors, and they're also actively communicating with the environment in the electromagnetic spectrum. This is an antenna that we, surprisingly enough, call an antenna. And the only difference between this antenna, well, there are other differences, but the major difference between this antenna and this antenna is the wavelength that they pick up. So they are both communicating in the infrared spectrum, in the electromagnetic spectrum. So when you take that moth antenna and you put it under a microscope, this is what those antennae look like underneath a microscope. And now, if you take one of the smallest hairs that you see here on these antennae and you put that underneath the microscope and you amplify it even more, this is what that looks like. And so it's amazing where we actually have, it's almost like fractal geometry where we have antenna layered on top of antenna. And if you look at this picture very closely, you can see very small hairs that are on the surface of this antenna as well. So it's a pretty profound picture. So as we have worked with many different growers, producing a broad variety of different crops in different types of environments, different diseases, we've observed a gradual evolution of plant health where plants become resistant to different groups of diseases and different groups of insects, based on what is happening inside the plant from a plant physiology perspective. So we put together this diagram called the plant health pyramid that describes some of the things that we see happening. And I want to be considerate of everyone's time. I've spent a bit more time than anticipated. So I'll give you a very quick overview. The foundation of the pyramid is when we observed plants are now photosynthesized completely, which means two things. One, it means that the volume of sugar production every 24-hour photoperiod increases anywhere from about 50 to 300%. And as well, the second piece that happens is that the plants begin producing more complex carbohydrates, more polysaccharides, and there are fewer monosaccharides in the plant sap. So at this stage, they become resistant to all the soil-borne pathogens, fungi such as Rhizoctonia, Verticillium, Fusarium, and so forth. And then the second stage is when plants begin producing complete proteins. They now become resistant to all the insects that have simple digestive systems, like chewing insects such as aphids and leafhoppers, all the larval insects, cabbage loop or tomato hornworm. So these first two levels are what we would refer to as passive immunity, where we're simply removing the food source for the potential pathogens. And then the third and fourth level, when plants begin producing higher levels of lipids, plant fats and oil, and secondary metabolites such as phytoalexins and terpenoids, sesquiterpenes and bioflavonoids. These are all compounds that plants produce in higher concentrations. This is now active immunity. When we trigger the SAR pathway and the ISR pathway, we get a very strong and very pronounced immune response that can actually kill disease and insect pests. And it's a lot of fun when that happens. That can happen very well. So when we think about management patterns, thinking about plants systematically in this process and trying to eliminate the root causes of why we have insect pests and disease pests showing up in the first place this is the place where using biostimulants is exceptionally powerful, because biostimulants can really accelerate the development. When biostimulants are combined with nutritional products, they can really accelerate plant health in this direction and help us produce active immunity very, very quickly. I would like to also mention for just a moment the impact of synthetic chemistry and the idea of protein synthesis versus proteolysis. So I spoke briefly about the capacity of plants to develop complete proteins versus incomplete proteins, having high levels of nitrates and amino acids in the plant sap. And I find it very interesting that whenever we apply synthetic pesticide, such as an insecticide or fungicide or so forth, it can actually switch a plant from protein synthesis to proteolysis. So it can trigger increased insectability. So often when we begin working with growers who have a desire to reduce pesticide use, we do not advocate that we immediately stop all pesticide applications. We have to earn the right to discontinue the use of pesticides. But I think it's very important when we want to begin working with biostimulants and to begin using predictive management, it's very important that we, if at all possible, we avoid the use of insecticides and fungicides prophylactically. We avoid using them as a defense mechanism or before there is an actual problem present, because it can actually have the opposite effect and can increase susceptibility to the problem that we are trying to avoid. This was first outlined by Francis Sciboso in a book titled Healthy Crops, which is readily available online. And Francis does a great job of describing protein synthesis versus proteolysis. There's been a lot of additional research since, and many new things that we have learned about plant immune systems. But some of these foundational ideas about the integrity of the compounds the plant is producing are still very valid and still hold true for disease and insect resistance. So the pathways to success, how do we put it all together? And how do we get the greatest response from biostimulants? One is we need to manage as an integrated system. And really try to use predictive and preventative management because that's where biostimulants will fit the best. And we need to try to reduce and avoid using rescue chemistry as much as we can because they generally tend to have a very antagonistic effect to the biostimulants that are in the system. And in addition, one of the things that we've observed is that application timing and consistency is absolutely critical with biostimulants. It has a much bigger impact with product performance than the typical nutrient applications like a potassium or phosphorus application would. One of the pieces that we've observed is that the difference between average performance and exceptional performance with the biostimulants is management intensity. Growers who are in the field, who are paying very close attention to what's happening with their crop and are trying to position nutrient and biostimulant applications for the greatest possible crop response. They're out there constantly, every day, every couple of days, monitoring and managing what's going on, rather than spacing out applications to be several weeks or more apart. So with that, I've taken a bit more time than I anticipated, and I want to be considerate of all of your time. So I'd love to open it for your questions and comments and feedback. Any thoughts that you have, feel free to write in your questions, and I'll go through and respond to those. So we have one question. So one question from Isaac is, we not concerned that Trichoderma would compete with arbuscular mycorrhizal fungi? So are we concerned that there would be potential competition between Trichoderma and Mycorrhizae? The answer is yes, they definitely will compete, and we think that that's a good thing. Not only will they compete with each other, but in fact, they also have the potential to have a synergistic relationship with each other. So it's not a question of either one or the other. There isn't a question that plants will selectively feed mycorrhizal fungi at the expense of Trichoderma or vice versa. Trichoderma are very aggressive and they can grow very quickly, but all the experience we have had with the two in the field, all the observations that we've had, is that they actually have a synergistic effect rather than an antagonistic effect. And we have not yet observed Trichoderma to outcompete Mycorrhizae to the point of causing a problem. So that may happen on some operations, but we haven't seen that at this point. So Carl asked a question, Carl Sapp, are you testing these in a greenhouse first? What is the process for figuring out what is working and what doesn't? So Carl, I'm not exactly sure of the context of your question, or when you say, are you testing these in a greenhouse? I'm not sure what these is referred to. So if you could ask that question again, I would certainly welcome any clarification. I'm assuming that you may be referring to what we described as synergistic versus antagonistic relationships, and the ways that we have identified these potential synergistic or antagonistic relationships is by a combination of obviously reviewing the scientific literature and reviewing the work that people have done, and then also combine that with actually trying it out in the field. So I think it's very important microbial biostimulants and understanding how biology impacts what is happening in plants. I believe it's very important to try that out in the field and not in a greenhouse, because biology behaves very differently in a greenhouse than it does out in the field, even if you have topsoil as the substrate. But particularly in soilless media, these biostimulants behave very differently in the greenhouse than they do in the field. So we actually do almost all of our research work where we're evaluating different materials and their compatibility out in the field and looking at what's happening in real topsoil. So that's a great question, though. Thanks for asking that. Steve Diver. Hi, Steve. A question, how are we doing with organic cucurbit and tree fruit production in the humid East, Ohio, Kentucky, and Pennsylvania, and which have particularly difficult pests and diseases? Do they ever? You're absolutely right. It's a challenging crop set to work with, and however, I would say, so with tree fruit, we actually find that tree fruit are some of the most responsive crops to balanced nutrition and biostimulants. They change very, very quickly, which has been quite a surprise for us, because I would have expected tree fruit to take longer to respond. But and I don't know exactly why they are so rapid. Perhaps as they have accumulated energy within the trunk and branches that they tap into for a much faster turnaround. I can only hypothesize as that at the moment. But we find that tree fruit respond very, very quickly, and that we can quickly produce very strong disease and insect responses to most of, I would stop short of saying all of the major tree fruit diseases. So fire blight, we've been able to produce resistance to very quickly. Apple scab, we've been able to produce resistance to very quickly. Sooty apple blotch, cedar apple rust, and so forth. We also have been able to produce resistance to fairly quickly. Powdery mildew is pretty easy. But then when you get to the insect pests, codling moth is one that is still a challenge. We have a few farms that have produced some very nice responses, but not enough consistently that we can say we've really figured it out. And the same would hold true of plum curculio. With that being said, actually, let me also speak to cucurbits for a moment. Cucurbit crops are, I would say, perhaps the most challenging crop of all. Primarily because, for whatever reason, they are pathetic at absorbing nutrients from the soil profile. Even when we use mycorrhizal fungi and we use bacterial inoculants, and for many of the cucurbit crops, our most successful programs, the growers that we're working with, are applying mycorrhizal fungi and bacterial inoculants combined with biostimulants every week to try to sustain and develop and grow the largest root systems possible. And even with that, so we're able to produce resistance to powdery mildew and downy mildew and so forth, the mildew diseases, quite consistently. We've had a lot of success there. But it's been in the last year that we've started having success with squash bugs, and cucumber beetles. And our biggest challenge has been getting these crops to absorb enough phosphorus from the soil profile. In soils where we have a generous phosphorus supply, we find that these plants, through the breeding or for whatever reason, even when we're applying all these inoculants and phosphorus solubilizers, even on completely conventional crops where we are applying soluble forms of phosphorus, these plants do not do a good job of absorbing phosphorus, and that is the critical missing link that is associated with many of the diseases and insect pests that they're susceptible to. In the last year, we have had some very interesting and unexpected side effects from a combination that developed. We will be testing it more extensively in 2018. But we've discovered that there was a combination, foliar application of Rejuvenate, which is an exceptionally strong bacterial biostimulant, combined with seaweed, combined with boron, that, well, what I can just say very simply, we trialed it in about 20 farms in 2017. So we still need to do a lot more research on it. But on those 20 farms, we achieved perfect 100% control of potato beetle and Japanese beetle and squash bug and spider mites. Every single pest that we tried it again, we achieved perfect control. So we're actually quite excited about that. And about that combination. There obviously are some synergistic effects there that we haven't observed before. And that's something that we want to begin experimenting with a lot more in 2018 and see if we can develop a product around that has some interesting side effects. So It's a great question. Those are some of the most challenging crops that we work with. Marie Omlin asked the question, star triggers in seaweed. Are the inspiring example with wireworm, are there any similarities with brassicas? And so we think of brassicas. Brassicas, the primary insect challenge there with corals, obviously there's wireworm and then also the cabbage looper. And we find that there is another, I was intending to mention this and I failed to earlier. There is another video clip online that you can find on YouTube where I talk about the plant health pyramid, and there's some additional information available on the AEA website. And there's some new things that we've learned that we need to add to this. But when we are successful with level 1 and level 2 of the plant health pyramid, which is purely chemistry, you can produce level 1 and level 2 in a hydroponic system by addressing a handful of, I think it's level 2 combined is 7 minerals altogether. When plants have added levels of those 7 minerals and those 7 nutrients, they will be resistant to all of the insects and the diseases that fit into those 2 categories. So for level 1, increasing photosynthesis, the nutrients are magnesium, manganese, iron, and nitrogen. And for level 2, the nutrients are magnesium, sulfur, molybdenum, and boron. So when we address, obviously magnesium fits into both categories. When we address those 7 minerals, we found that we get an exceptional degree of resistance to all the insects that have simple digestive systems. Tom Furman asked the question, what does Azotobacter need to be successful if not a synergistic relationship with a legume? So that's a great question. And it is exactly the same thing that all bacteria, phytosanitary bacteria on the leaf surface or mycorrhizal fungi need to develop a strong population of soil system. And that is, they need sugars from the root system as root exudates. They need a food source. And I think one of the things that we have done well at AEA is with our use of foliar applications, we have focused very intensely on making sure that plants are constantly photosynthesizing really well and producing a large volume of sugars. So when you have a large volume of sugar production and the sugar production moving out through the root system as root exudates, you are now really stimulating the Azotobacter and the plant growth promoting rhizobacteria and all of the beneficial bacteria that are in the root system. And that's why we're producing such a strong nitrogen response from Azotobacter, I believe, is because you're giving them an abundant food source. It's a great question, Tom. And one final question is, can I elaborate a little bit more on bitter pit issues? So bitter pit is a whole combination in and of itself. Actually, I think the most recent webinar, or the second to last webinar that I taught was titled How to Manage Nutrition in Stone Fruit. And I spoke a bit about I spoke a bit about bitter pit and calcium-related physiological disorders. So I would ask you to go back and look at that if you want a more in-depth explanation. But what I can say very briefly is that calcium-related physiological disorders in fruit have nothing to do with a calcium deficiency, or I should say they have nothing to do with an inadequate calcium supply. It's not about having inadequate calcium supply. The problem is that during the cell division stage, so after we have a 10 to 14 day window of division. In that 10 to 14 day window, calcium and potassium have an intensely antagonistic relationship at all times. But in this window, it becomes very critical. And what happens is that potassium wants to move into the embryo, and it prevents calcium from moving in. So the goal is that we need to keep potassium levels at the cell division stage inside the plant very, very low to allow calcium to move in. How do we do that? It's relatively simple. One is we stop the potassium applications early. We wait until we get to the fruit fill period. Step 2 is apply levels of manganese to serve as a potassium thermostat, potassium regulator. 3 is we apply generous levels of boron so that we have good calcium mobility. And 4 is we apply generous levels of calcium. So it's really those things. Stop potassium, add manganese, boron, and calcium. And you can resolve bitter pit, blossom end rot, bitter rot, brown rot in peaches, almost essentially all of the calcium-related physiological disorders in fruit are generated to excess of potassium that is creating a calcium deficiency. So, and you can find more information about that, more about how that works in the recording, which is available on YouTube if you want to look at that. All right, so I believe I've answered all the questions. Thank you very much for joining us tonight. I hope that you found this information helpful and useful. And please join us on any future webinars and contact the AEA team. You can reach our team at advancingecoag.com and call us if you have any further follow-up questions. Thanks for attending tonight, and we'll talk with all of you soon. Be well. Bye.